Okay, so we're going to talk about a couple specific type of enzymes in this last lecture. So one of them will be lysozyme. And so lysozyme is an enzyme that will hydrolyze peptidoglycans or chitin with 10 to the 8th fold rate enhancement over non-catalyzed reaction. And so this enzyme right here will bind this sequence right here. So you can see this peptidoglycan that binds into this active site. So we talked about this little cleft, and it will cleave at this position right here. And so we want to take a little bit closer look at that. This was actually the first crystal structure that was solved. It's typically a small enzyme, 129 amino acids, only 14.3 kilodalton. Fairly rigid, it's cross-linked by 4 disulfides. The smallest substrate that it'll actually process are hexasaccharides. So if you have anything less than a hexasaccharide, it may bind, but it won't really process it. And so the non-reducing end is at A, and F is the reducing end, meaning this is where I'm going to have my hemiacetal. You can imagine that it's kind of in that direction there, if we think about the normal orientation of drawing a carbohydrate. Now, to get this structure, they didn't want it turning over, so they actually bound this NAG3, N-acetylglucosamine. So that is bound in this ABC site. So it was actually crystallized with that bound. And then D, E, and F were inferred by modeling. And so the cleavage site is actually at this part here. So to get this crystal structure, they found out that this little fragment will bind. That would help stabilize it, allow them to get a crystal structure. And then they modeled in these additional residues. Now, actually, I want to go back. If you notice in the structure right here where the cleavage is, notice how they actually drew out this 6-membered ring. Notice it's actually in a half chair, and that's actually significant for this enzyme processing. So the residue D, and again, sorry to keep going back, but that's going to be this one right here, and the cleavage is going to happen right there. So residue D, and actually you can kind of see it in this picture if you squint really hard, that actually has kind of a planar structure. So sugar residue D can't bind well without changing its conformation. And so it changes its conformation to kind of a half chair. And that helps this mechanism. And so we know where it cleaves because you can do this reaction in labeled water. So it's labeled with oxygen 18, and you can see that the water actually attaches to that, what they refer to as the D saccharide, and cleaves between position D and E. So it lets us know the exact position of that cleavage. So here is the modeling of the amino acid residues with the backbone of just this hexasaccharide. Now this is oriented in the opposite direction than those previous pictures. They come from different authors, but anyway, so now we're looking at it running the opposite direction. Here is the reducing end, and this is the non-reducing end. And we're going to cleave in this position right here. And notice that this has to adopt kind of a half chair conformation because of the residues that are around there. Now, typically you have N-acetylmuramic acid residues with a lactate here. And that lactate's what allows this to bind to proteins. Remember when we talked about peptidoglycans? And so the only positions that will allow that lactate residue here are positions B. So I could have that lactate here, or D. I could have it here, or F. So that gives you some registration of where this is going to bind. You're only going to be able to bind when that lactate is at those positions there. So lysozyme reaction is conversion overall from a hemiacetal, or sorry, from an acetal to a hemiacetal. So this is the reverse of the reaction that we talked about in carbohydrates, where we wanted to form an acetal. We're going in reverse. And so in short, what we can do is protonate one of those oxygens. That makes a good leaving group. You can think of these electrons collapsing down and kicking that out. That gives you this oxonium ion right here that is in resonance with this carbocation. So we got two different resonance structures there. And then what's going to happen is water is going to come in and either attack that carbonyl and kick those electrons onto the oxygen, or think of it attacking that carbocation there. And we deprotonate it and we end up with a hemiacetal. The whole key here is that we're going to hydrolyze a carbohydrate chain. We're going to separate two carbohydrates from each other. So the non-enzymatic reaction is acid catalyzed. So we did this all the time in organic chemistry. We hydrolyzed acetals by adding in a little bit of acid in water. In the enzymatic reaction, it includes a residue to act as a general acid. So that acid is going to be supplied by a amino acid side chain, and you're also going to stabilize the transition state. And so here is a mechanism. This is out of a different book, so it looks a little bit different than what we do, but they show potential mechanisms either through an SN1 mechanism, where the leaving group's going to leave behind a carbocation, or through an SN2 type mechanism. In both cases, you're going to have glutamic acid here at position 35 that acts as the acid. And so what's going to happen there is you're going to get protonated. So essentially this bond here breaks and you get protonated at the same time as those electrons collapse down. So just to connect it with the mechanism that we saw before, what they're really saying is as this comes out and grabs the H, these electrons collapse down. So kind of a concerted mechanism, and that would get you to this state right there. So imagine that as these electrons come down here, I'm coming out and I'm grabbing a hydrogen there. The other thought is, well, I could actually get the aspartate here to come in and attack that carbon there and kick out those electrons and pull off that hydrogen here. We're going to get to the same point. If I go through this SN1 type mechanism, I still have an aspartic acid residue that will come in and attack that carbonyl, or sorry, that carbon here, or this oxonium ion. Or in this case, if this came in and attacked, then I've got this covalent intermediate. And again, that bottom step there is this reaction here, where water is either going to come in and attack there and kick those electrons up onto the oxygen, or come in and attack here. Oh, and no kicking out because carbon only has 6 electrons. Now, if you notice, this transition state right here is planar. And so if we could stabilize this, or if we go through this mechanism here, and it does require this transition state, then that would kind of suggest that it goes through this SN1 type mechanism. Let's just kind of finish up the mechanism. So on the right side here, if that aspartyl didn't attack, then we need to get water in there. And so how we're doing that is the deprotonated glutamic acid is now going to deprotonate water. That's how the glutamic acid is going to get its H back, and then the water would attack that carbonyl. Or in the other case, we have this aspartate, and so now that could serve as the leaving group. Same thing, the glutamate is going to deprotonate water, and then the oxygen is going to come in and attack and kick out that aspartate. And so essentially in this case, we're kicking out the enzyme as the leaving group. Ultimately, we get to the same place. We get back to our protonated glutamic acid and our unprotonated aspartate, and we have cleaved our carbohydrate there. So here is a little bit better idea, or picture, maybe not better, a more detailed, there we go, detailed picture of the residues there and the roles that they are playing in helping orient the saccharide or the oligosaccharide so that we can actually cleave it. So binding to the D spot, we strain it to a half chair, and that is helped to be stabilized by this valine at position 109. if we actually mutate this valine to a proline, we actually inactivate it. So we know that that valine is actually important there, because changing it will cause inactivation of the enzyme. And what else we know is that it does go through that planar transition state, because these molecules here where we have this lactone, again, that's planar, ends up being a good inhibitor. It binds tightly to the D site there. So if we have this at that position, it will bind and inhibit. Which lets us know that we are actually likely going through this type of transition state, a planar transition state. Now, there is some other evidence for that. We can actually get some covalent modification. So the covalent intermediate is short-lived, but they did analyze it. So if we go back here and we're trying to figure out which one of these enzymes is it, it looks like it's kind of a mixture. We do get this planar transition state, but there is a covalent attachment to aspartate. And I didn't set that up very well, but that's why we're looking at these two different mechanisms, this SN1 and SN2. These are the two possible different mechanisms, and we're trying to figure out, well, how does the enzyme really work? Well, from the first set of things that we just talked about, yeah, it does seem like it goes through this SN1 because it does require this planar transition state. But there is evidence that we do get a covalent attachment. But as I just said, that covalent attachment is very short-lived, but they have shown it. And actually, if you mutate it to that position 35 to the glutamine, it actually slows down the acid-base catalyzed step. Now, what you can also do is take the substrate, and what you can do is put some fluorines. Now, fluorine is going to be a decent leaving group, and that's going to be a good leaving group without acid catalyst. And actually putting this fluorine here is going to destabilize the transition states on both sides of the intermediate. And so what that means is it's going to favor the tetrahedral, or sorry, not the tetrahedral, well, yeah, it's tetrahedral, but the covalent adduct there. So what they've done here is the yellow is this tripeptide non-covalent binding position. And so you can see it in yellow there. And then this molecule right here, which is this covalent inhibitor. So it binds to that aspartate residue. And you can see that right here, it's bound to the green, and it's a covalent, and it permanently binds. And so if you look at that, there is a lot of overlap between those. Anyway, suggesting that you do get a covalent attachment to that aspartate, but it's a very short-lived one there. And so really we are going through kind of this planar transition state. And so here is just another view of this covalent attachment of this inhibitor that lets you know that the aspartate is close enough to form a covalent attachment to that anomeric carbon right there. Okay, so lysozyme, its rate is enhanced by acid catalysis to assist in the bond breakage. So we actually have a proton there that helps protonate that acetal. We get stabilization of the transition state. So we make that sugar adopt a planar conformation, and that aids in the bond breakage. We do get covalent intermediate formation that helps stabilize it. And again, stabilization of the transition state conformation aids in the bond hydrolysis. So that is lysozyme. And let's look at some other enzymes such as serine proteases. And so serine proteases in there, what they are, well, let's talk about proteases first. They rapidly break peptide bonds, but they can also break ester bonds. And you can imagine that they would hydrolyze an ester bond faster than the peptide bond. If you remember your reactivity of carbohydrates, or not of carbohydrates, of carboxylic acids and their derivatives, esters are much easier to hydrolyze than amides, and that's due to the donation of the electrons from the nitrogen that actually really stabilizes that bond. So proteases are going to be things that break down proteins or hydrolyze them. And like I said, they can hydrolyze esters as well. Now we're going to talk about a specific type of proteases, those known as serine proteases. And the key is that they have a conserved serine residue, and the serine is going to act as the nucleophile. And so there are some examples: chymotrypsin, trypsin, elastase. These are digestive enzymes that bind and cleave at different sequences and peptides. So you can imagine if we want to digest protein, we need to break peptide bonds. And so these are going to be enzymes that actually break down peptides. Now, I'm going to give you a little bit of a conundrum here. Well, if these are peptides here, or proteins, and they digest proteins, won't they digest themselves? Can a chymotrypsin cannibalize another chymotrypsin molecule? Interesting. So, serine proteases, they act by covalent intermediates. And we know that because we can take molecules, kind of like what we did before with lysozyme, and we add them in with these serine proteases, and they bind irreversibly. And so we've got this molecule here, diisopropyl phosphofluoridate, known as DIPF. And if you introduce it into chymotrypsin, it will bind irreversibly. And so that gives us kind of a hint that and then chymotrypsin doesn't work after that. So now what we refer to is a dead enzyme. So that kind of lets you know that that serine's important, or at least covalent attachment to that serine shuts down the enzymatic activity. Now, these serine proteases are very important in nerve cell modulation. So acetylcholine is a neurotransmitter. And if you have too much acetylcholine, what happens is you are overstimulated, and essentially you get muscle contraction that is irreversible. And after a while, you'll use up all your ATP, and essentially you just die from kind of muscle fatigue. If this happens in the lungs, you can paralyze the lungs, and so then you can't inhale oxygen. But typically these are neurotransmitters, so they react in your brain and just kind of overstimulate your muscles, and your muscles are just going to be permanently contracted, and eventually you die. So we use these as insecticides. Parathion, you may have heard of that, or malathion. These are insecticides. These are similar to glyphosate that's actually used to kill weeds. So they all have this very similar structure. So these kill insects, and this kills humans. So sarin gas is a very potent inhibitor of acetylcholine esterase. So I didn't really finish that. So this is a nerve transmitter, but you don't want too much of it. So we actually have an enzyme called the acetylcholine esterase that breaks it down to choline and this acetate group. And by doing that, you stop the nerve impulse. Now, if I inhibit this enzyme so I can't break down my acetylcholine, when you release acetylcholine into your presynaptic cleft, you're just going to overstimulate those, and the enzyme can't break it down. So these molecules here all inhibit the acetylcholine esterase and cause death, this of insects and this of humans. In fact, there was a recent assassination of Kim Jong, the leader of North Korea, his brother, I believe, or some relative. And what they did is they put some sarin on a glove, and these two girls jumped the guy in an airport, and they rubbed it on his face and I believe the back of his neck, and that was enough to kill him. There was a cult in Japan that released sarin on a subway station. They did a poor job of releasing it, and so the death toll wasn't as high as it could have been. I shouldn't say a poor job, but they were very inept in how they did it, which was good because this is a very potent neurotoxin. Anyway, I can't remember how many people died. There's the bonus for this lecture. How many people died in the sarin nerve attack in Japan? But yeah, not nearly as many died as could have. So horrible experience. These are essentially outlawed as chemical weapons, but they're still around. It was thought that Iraq had these. That was one of the reasons that we invaded them, is they had these weapons of mass destruction, such as sarin. Anyway, and here's the other molecule that we looked at, the binds. So that's so fun to talk about, killing people with nerve agents, but let's talk about serine proteases. So another clue about the mechanism is we have this molecule right here, TPCK, and what we have is a leaving group. And so we actually can attack here and kick out this leaving group. Now, chymotrypsin cleaves the amide after a hydrophobic residue. So if this was, imagine a nitrogen and the remaining part of a polypeptide, it would cleave this site right there. Now, it's got this hydrophobic part, and when you introduce it to chymotrypsin, chymotrypsin actually covalently is attached to that carbon right there. That lets you also know that there's a histidine that is in close proximity to the site of cleavage there. And then there is another enzyme, trypsin, and trypsin cleaves after lysine. So again, if we play this trick and we put kind of a lysine residue there, but we don't have an amide, we've got this leaving group, we can form a covalent adduct with histidine using this molecule right there. The whole take-home message is we can get these covalent adducts, and it lets us know what the active site looks like, what residues are possibly playing a role in the reaction. And so what we find in serine proteases is, again, the serine's conserved, but we also see a catalytic triad, which is an aspartic acid, a histidine, and a serine. And they end up in this general structure right here. And so you end up with this catalytic triad. Here's your serine protease. And then what determines the specificity of these enzymes, where chymotrypsin will bind hydrophobic and trypsin will bind lysine or cleave after lysine, is the specificity pocket right here. So let's take a little bit closer look at some other serine proteases. So like I said, chymotrypsin will bind bulky aromatic side chains. And so if you look at the specificity pocket here, what you find is that it really has some small groups, glycine, and of course you've got a serine, well, sorry, this isn't the active serine, but you do have a serine down there. You also can cleave after tyrosine, and this probably helps assist with cleaving after tyrosine. Anyway, your actual serine protease is going to be doing its work up here. Now, trypsin, like I said, cleaves after lysine. So in this specificity pocket, what you have is an aspartic acid. Now, lysine's positive, and the aspartic acid is going to be negative, and so that is going to select for residues that have this lysine, and then we're going to include that bond there. And then there's an elastase, and it cleaves after small neutral residues. So in this pocket, what you find are some bulky residues that really don't allow much specificity here. So, or the specificity is this group has to be small. Now, if all I do is mutate in trypsin this to a serine, so essentially I change this group to serine, it doesn't make it so that trypsin will cleave like chymotrypsin. So there's other groups that are important for that specificity, but this pocket is one of them that provides that specificity. Now, if I change other residues in this pocket, I can make it so that trypsin does cleave like chymotrypsin, just not as well. So it seems like there's some other specificity that's going on in this pocket, and so I need to kind of match that up. Okay, here's just another picture of it. So chymotrypsin, essentially just a hydrophobic pocket that will bind large hydrophobic groups. Trypsin has the aspartic acid that will bind lysine, and elastase has bulky groups that will only allow the binding of small peptides in there. Now, what's really interesting is if you go and look at these serine proteases and you try to see how related they are, you'll realize that they're not really related. And we can look at this by the position of these residues. You can see that the position of these residues really aren't conserved. And like I said earlier, the active site residues can be spread all across the backbone of the polypeptide. And so what you find out is the family of serine proteases containing trypsin, chymotrypsin, and elastase is older than the divergence of prokaryotes and eukaryotes. So this was an enzyme that was found to be useful long before we got prokaryotes and eukaryotes. There are other families of serine proteases that their primary and tertiary structures bear no discernible relationship to those of the trypsin family, but they still have the same catalytic triad. And so that's really interesting that this catalytic triad has found its way into these serine proteases probably through several different routes. And what that just means is that is a very efficient arrangement of amino acid side chains where you can actually cleave these peptide bonds and do other chemistry. So here's an example of that catalytic triad. And so again, everything that has a catalytic triad does not have a common ancestor. And so here is the mechanism of the serine protease. And so what you do is you have that histidine that we know is important there, and it is in complex with that OH in a hydrogen bond. And essentially what it's going to do is act as a base and deprotonate that serine, making that serine much more nucleophilic and allowing it to come in and attack that carbonyl, forming a tetrahedral intermediate. And then that tetrahedral intermediate can collapse and kick off this nitrogen. And when that nitrogen leaves, it needs to pick up a proton so that it is not negative. And so that leaving nitrogen abstracts that hydrogen right there. Now we've got a covalent adduct between that one half of the polypeptide and that serine residue. And I need to get rid of that serine residue. So now water will come in again. And so you can see that in this case, histidine is going to act as a base. And we pull off that water molecule there, that hydrogen off that water molecule and allows attack. We get a tetrahedral intermediate that is going to collapse down and release the serine. And again, we don't want to release an O-. That O- pulls the hydrogen off of that histidine. Now, if you notice, I really didn't talk about the spartate. So the spartate's action's happening kind of in the background or on the back side of the histidine there. So histidine, if you think about it, it's got this basic site over there. And what we need to do is be able to make it so that we stabilize this histidine when it is protonated. And so this aspartic acid there helps coordinate that hydrogen and will help stabilize this is positive by pulling that nitrogen away. Anyway, essentially it just works kind of in the background there with the histidine and complexed with that that hydrogen on the backside of the histidine. Okay, here is just another example out of another book, so you can walk through it there. Something that this one really doesn't show, but we'll talk about now is there is what's referred to as an oxyanion hole for these tetrahedral intermediates. So when I form that tetrahedral intermediate, I need some place to park that negative. And so what they see in these chymotrypsin is a pocket that they call the oxyanion hole that can help stabilize that O negative right there. So again, that oxyanion hole, you can show that it's important for stabilizing a negative. And so again, we kind of have a covalent transition state or tetrahedral intermediate. So if we attacked a boron there, we would get this tetrahedral intermediate, and it parks that oxygen into this anion hole right there. So when you're reading about the oxyanion hole, that is it. So in serine proteases, we get preferential binding of the transition state, and we can actually enhance the rate of cleaving a peptide 10 to the 10-fold. So you're 10 billion times faster than just in water. Now, what's interesting is if we actually remove any of those catalytic triads, we can actually still get a rate enhancement. It's not nearly as high, but we can still get a rate enhancement. So I'm going to leave you with that as an interesting question. So as you can imagine, this reaction is very dependent on pH because we're abstracting and adding protons. And so they can actually get crystal structures of these different intermediates and complexes by getting crystals at different pHs. So if you crystallize this out at a pH of 5, you actually get the acyl enzyme complex. Histidine 57 is protonated and acts as an H-bond donor to water. If you get the pH and you raise it to 9 and then you freeze it, the histidine gets deprotonated and triggers the hydrolytic reaction. So just to let you know that they do some tricks to understand the mechanism, and crystallography can reveal some insights, but typically crystals are just capturing a very homogeneous state of the enzyme. So NMR is very good at probing mechanisms, but you can also do it in crystal structures. You just have to tweak the conditions a little bit to get some mechanistic information out of them. Now, I asked you an earlier question about these trypsin and chymotrypsin. These digest proteins. Well, if trypsin and chymotrypsin and all these other ones are proteins, why don't they digest themselves? Well, it just happens that there are inhibitors that will bind in there. And so this is an inhibitor that's actually a protein. It is called the bovine pancreatic trypsin inhibitor. It's a 58 residue protein, binds with very high affinity, and essentially prevents the bovine trypsin from digesting the pancreas. So if you're making these enzymes, another question is, well, why don't they digest the cells and everything around there? And why aren't they digesting themselves? Well, they tend to be inhibited, and they're only activated at the site of action. And so in this case here, the green part is the inhibitor. And what happens is you form a very, very tight interaction between the inhibitor and trypsin. And in fact, when you crystallize this, you can see that the serine is actually in coordination with that carbonyl there. But the binding of this is actually so tight that the reaction can't proceed. So essentially, this is like a transition state inhibitor, the green portion there. It essentially inhibits the processing of that serine and hydrolyzing that amide bond. So again, to answer that question, well, how do we prevent ourselves from digesting ourselves? Well, we produce these trypsin and these chymotrypsins as inactive enzymes. And those inactive enzymes are referred to as zymogen. So zymogens are these inactive precursors, and they're activated by other proteolytic enzymes. So other enzymes that cleave proteins. So one of these is the enteropeptidase, and it activates trypsin. And actually active trypsin will also activate trypsin. So what you do is you synthesize the trypsin distant from the site of action. It's transported to the site of action, and there you have an enzyme that will act on these zymogens, cleaving off a group, forming the active enzyme. And so the zymogen in this case is trypsinogen, and then you have the enteropeptidase or trypsin. And so you can imagine, oh, I got trypsinogen and one enteropeptidase is cleaved off that group, and I now have active trypsin. Well, that active trypsin can then activate more active trypsinogen. This is a process that we use in blood clotting, very similar. I'm not even going to try to touch this, but what's really cool about this is if you activate one group and then that group can self-activate itself, you can actually get what's referred to as some cascades. Now, blood clotting is a very intricately controlled cascade. I mentioned this in an earlier lecture. You don't want your blood coagulation to just occur willy-nilly. the way that it works, if all of a sudden everything just went haywire and all of a sudden your blood coagulated, you'd be dead. So you want it to occur at sites of damage, but you don't want it to occur at an uncontrolled rate, but you do want it to occur fast before you bleed out. So I know that sounds very contradictory, but just think of all the factors that need to be involved in blood clotting to make sure that it is effective. And you can see that actually it is a very intricately controlled process, but you do get these cascades where you do get activation of one enzyme that can then activate similar partners like itself. And that means that you can get a response really fast. Once I activate a protein, it can activate other partners like itself, and you can get quick responses. Okay, I'm just going to leave you off that there's other classes of proteases, other things will cleave peptide bonds known as cysteine proteases. These are known as caspases, cysteine active proteases. They're involved in tumor and apoptosis. You got aspartyl proteases, and you also just got metalloproteases. So we're going to leave it with that. Here's some checkpoints, and we will catch you in the next lecture.